All right, I think we're live, so let's get this thing going. How's everyone doing today? My name is Andrew Erickson. I'm a director at Allele Capital. We at Allele Capital work with the Soligenix team and are helping kind of put on this event today. So appreciate everyone for dialing in. I'm joined by Dr. Shaber and Dr. Poulion, who are going to spend some time today speaking about who Soligenix is, what this investigator-initiated study they are working on is, and more importantly, how that leads into their confirmatory phase 3 study for their lead asset, HyBryte, which is targeting CTCL, which we'll dive a little bit into as well. So before we dive in, for those of you who are unfamiliar with Soligenix and HyBryte, HyBryte is their lead asset, which is a topical therapy applied to cancerous lesions on the skin, which is activated by visible light. So we're going to get more into those details as well as the lead indication in CTCL. Before we do that, I'd love to kind of kick off the call with some introductions from our speakers. So Dr. Shaber, maybe we can kick things off with you, providing a bit of a background.
Of course, Andrew. Thank you for having me, having us. My background, it's quite long. I've been in the industry for 35 years, started in big pharmaceutical companies, and then moved into small biotech, where I co-founded a company of my own back in the mid-90s that I had successful exit from in the mid-2005 range, and then joined what is now Soligenix to build out the pipeline for the company and really build out the company. So it's been quite a journey, and as we're going to touch upon, we have a lead program here that's very exciting.
Fantastic. Thank you for that. And Dr. Poulion?
Hi, yes. Hi, everybody. My name is Christopher Poulion. I'm a physician by training. Been with Soligenix now for almost 15 years. I'm the medical director here. In my capacity as medical director, I'm responsible for assisting the company in all manners of our clinical development programs, including but not limited to protocol design and implementation, the execution of the clinical study protocols, safety review, and pharmacovigilance. I work with our contract research organizations to help with all the logistical aspects of the clinical trial. And specifically because we focus so much on orphan and rare disease, it's very important for me to have a good understanding of the medical landscape itself. And we're going to talk about cutaneous T-cell lymphoma today. And that medical landscape is comprised of patients being treated specifically at specialty centers. These specialty centers are household names like the University of Pennsylvania, Stanford, Mayo Clinic, MD Anderson, etc. And one of the core responsibilities that I have is to go out and to meet with all these different leading experts at those institutions and to make sure that the research that we're conducting incorporates their feedback so that I can be sure that what we're doing is of the highest quality and also the most significant clinical relevance that we can bring to the patient community as a whole. We also interact frequently with the patient advocacy organizations, again, to help make sure that one, we're educating the patient community, but also to make sure that we're incorporating their feedback into our clinical trial design and development programs as well. I can honestly say that after working on this for now over a decade, it's essentially become my life's work. So I'm very happy to talk with you all today and answer any questions you might have.
Fantastic. Thank you both. Appreciate those backgrounds, and I think that helps kind of set the stage for our discussion today. So before we jump into the specifics of the investigator-initiated study and HyBryte and the phase 3 going on, maybe it makes sense for Dr. Shaber, for those not familiar with the Soligenix story, can you begin with providing a brief background of the company and overview of what you guys are focused on?
Of course. Soligenix is a publicly traded NASDAQ-listed company. We focus on developing and commercializing products to treat rare diseases in areas of unmet medical need. We have two areas of focus. We have a specialized biotherapeutic segment focused on small molecules and oncology and inflammation. And then we have a separate public health solutions segment focused predominantly on heat-stable vaccines for emerging infectious disease and biothreats. And this segment of business has been funded entirely by the U.S. government to date, which has really allowed us to build out a more robust pipeline than you would typically see for a company our size and market cap.
Very nice. Thank you for that. Obviously a lot going on with the Soligenix story, and I think, there's multiple events we could probably do to cover all that. But as of the focus of today's webinar is being on your lead product candidate, HyBryte, which, for those not familiar, is in phase 3 development for cutaneous T-cell lymphoma, or CTCL, as we'll refer to it throughout the call. Can you please provide a short overview of the CTCL disease and how you are looking to treat it with HyBryte? That's for Dr. Shaver.
Of course. I have a slide. Maybe it'd be helpful to show the slide and I could run through the disease itself. So cutaneous T-cell lymphoma, or CTCL, is a rare class of non-Hodgkin lymphoma in which malignant T cells migrate to the skin surface to form patches, plaques, lesions, tumors in its most severe form. CTCL affects over 40,000 non-Hodgkin lymphoma patients worldwide, and there's currently no cure. And that's important to remember. Our therapy isn't a cure. You treat the disease, it goes away, and it comes back again, the same location or different location. So you really need to manage disease for the life of the patient. We estimate the market opportunity to be significant here, and it's really divided into two main subgroups, this disease. Mycosis fungoides, which is early stage disease, which is that upper right-hand picture in the diagram there. And that's where, luckily, the vast majority of patients are. The disturbing middle picture is known as Fusari syndrome. And as you would imagine, your prognosis for survival is not very good there. So luckily, our focus is in those majority of patients, those with mycosis fungoides. There's currently no approved first-line therapy for early-stage CTCL, which is very important. We have the potential, HyBryte has the potential, to be the first frontline therapy in treating this disease. So that's why it's so important, because it's an unmet medical need. And we're looking to focus on those vast majority of patients with mycosis fungoides, which makes up almost 90% of the disease population.
Gotcha. Yeah, thank you for that background. And obviously, tragic disease, and there's a severe unmet need for this rare disease landscape and rare diseases like this. And it's obviously exciting to see the support from the regulatory bodies with rare disease development and trying to push these types of therapies forward. So really exciting to see what you're working on and how you're approaching that. So appreciate the overview, and maybe now we just kind of jump right into the investigator-initiated trial. So jumping into that HyBryte clinical program, can you touch upon what studies have been completed to date and where you are in development in that process? I think that'd be good for the viewers to understand.
So that's a question that you're directing to me, I believe, correct?
Yeah, that'd be great from you, Dr. Pullian, and then
Sure.
Dr. Shaver, if you have anything to add to it, obviously, please feel free to jump in.
Sure, absolutely. So the investigator-initiated study, one of the key components of an investigator-initiated study to understand the difference between that and a sponsor by industry is exactly that. It's an institution-led study. It is often done by a clinician at a major academic institution, and it's less focused necessarily on specific drug development than it is on answering a specific question that's more germane to their medical practice. Oftentimes, what they may be doing is following up on other research that's been conducted by industry to get a better understanding of how they're going to be best able to treat their patient population. And that's exactly the case we have here at the University of Pennsylvania. The University of Pennsylvania, this is being run by Dr. Ellen Kim. She's the director of their cutaneous lymphoma program there. She was also the lead author and lead principal investigator on our FLASH study, the first successful phase 3 clinical trial that we conducted in this treatment landscape. She is also the lead principal investigator for our current ongoing confirmatory phase 3 clinical trial, the FLASH 2. Basically, what she's doing is she's treating in a very similar manner to what we did in that original phase 3 study. The patients are coming to their clinic and they're treating with HyBryte 2 times a week, and then they're getting exposed to the light therapy in the clinic. They do this 2 times a week, either until their disease clears or they have either until the disease clears or they reach a 1-year time point. And that's important because different than some of the registration studies that may be done, this is more geared towards real world use. Okay, you're treating the patient until the disease goes away, essentially, is the goal of this study. And we do have a slide that I think will help elucidate if you can show that explains why it's important in the context of our research. The HyBryte study, as you see there on this slide, we're treating them for 2 times a week, again, all the way up until a year. And the way we're measuring the effect there is we're using a scoring system. It's called the CALE score. That CALE score basically it takes the different index lesions, the areas of disease on the patient's body, and it takes into a bunch of different characteristics of those lesions, and it assigns a numerical value to them. Okay, and you do that at baseline when the patient first comes into the study. And then in the different efficacy time points along the way, you compare how those lesions respond, okay, to baseline. Now, this scoring system is something that is very known in the community. It's a validated scoring system. It's been used in the approval of previous therapies, and it is basically the gold standard for clinical research in this area. It's the same endpoint that we used in the first successful phase 3 clinical trial. The way we treat these patients when they're getting treated twice a week is we started an entry-level light joule dose of 5 joules, and we basically titrated up as best as the patient can tolerate. Again, treating for an entire year. It's open label, so everybody that's on this study is getting active drug. Everyone is receiving HyBryte, and everybody knows that. Now, I can talk about some of the response rates that we're seeing thus far, and you'll understand why they become important in a little bit in relation to the Flash 2 study. So, like I said, we're taking this CALE score, this classification of this composite score of lesions, and we're measuring it at different time points along the way. One of those time points is at week 18. Okay. Now, at week 18, we had 9 patients in total that were enrolled into the study, and 8 patients reached that time point where they were available for the week 18 time point. Of those 8 patients, 6 of them were treatment responders. The way we defined the treatment response was greater than a 50% reduction in their disease from when they came in at baseline. In the context of this landscape, that's actually quite quick. That's very nice to see. That's a very positive effect. Furthermore, in addition to that 75% response rate, what we saw is that 85% of the patients throughout the course of the trial thus far have received that time point and that success of greater than 50% response. And finally, 3 of our patients that are enrolled in that clinical trial are complete responses, meaning they're in remission of their disease. There's no more visible evidence of that disease on their skin anywhere. That is very successful. That's obviously something that's very positive. Dr. Kim and her team at the University of Pennsylvania are very excited. It's very good news for the patients. And finally, one thing to highlight, and this is true across our entire HyBryte program, but specifically for the investigator-initiated study, it's very well tolerated. The therapy itself is not loaded with a heavy side effect profile. The patients are able to come in and be compliant with the protocol. There's been no serious adverse events that have been related to the study drug at all. And so because of that, they're able to comply with the study. They're able to comply with the treatment. They're seeing the responses, and it just further motivates them to really continue to participate. Now, why is all that important in the context of the Flash 3 study that we're doing right now, the confirmatory trial? It's pretty simple, basically. If you look at this side-by-side comparison, you'll see that the IIS and the Flash 2 study, the phase 3 confirmatory trial, are extremely similar. I mean, in this situation here, basically what we're looking at is, yes, it is double-blind, randomized, placebo-controlled, as it should be. It's a controlled phase 3 clinical trial. But then when you look at the patients and the patient population that we're bringing in here, it's basically the exact same inclusion and exclusion criteria. Okay, so very same patient population that we treated in the investigator-initiated study and we treated in the previous phase 3 trial are in the Flash 2 patient population. It's the same scoring mechanism. So we're evaluating that same CALE score, that same primary endpoint in the Flash 2 study as we are in the investigator-initiated study and as we did in the previous phase 3 study. It's the same treatment modality. So they're following the exact same treatment schedule. They're putting the HyBryte on. They're coming into the clinic 2 times a week to receive their therapy. And we're tracking them for up to 18 weeks. And it is continuous therapy. just like the IIS. There's no treatment breaks. There's nothing else like that. They're treating 2 times a week, straight through, with us looking at safety and efficacy all along the way. The primary endpoint is at 18 weeks. And I'm going to bring it back to why I highlighted that 18-week time point in the investigator-initiated study. At 18 weeks, remember, we saw of the 8 patients that got there, 75% response rate. 6 of 8 of those patients following the same treatment parameters, with the same study drug, under the same conditions. Personally, in my opinion, I have every reason to be confident, given those different factors, that we should see nothing but positive results coming out of this Flash 2 study. Everything mirrors it so closely and so lined up that it'd be hard for me to figure out a reason why we wouldn't be optimistic about this clinical trial. And Dr. Kim said so herself. I mean, she actually just recently gave an interview with the Cutaneous Lymphoma Foundation that's out there publicly presenting very similar data time points. We currently have ongoing study, the Flash 2 study. It is ongoing. We started it in December. And I can tell you that the safety profile, currently what we're seeing so far in the Flash 2 study, mirrors everything we saw in the investigator-initiated study and the original Flash study. We've had no serious adverse events to date. I actually think I also have another background slide we could pull up that compares the original Flash study to the confirmatory phase 3, if you want to maybe segue there.
Yeah, that's a good transition, and I appreciate that detail with the IIS comparison in the Flash 2 study. And I was going to ask you, so good transition, if you could talk a little bit to these comparisons of that, that first phase 3 that the Soligenix team completed, and now how that compares to the current confirmatory phase 3 going on.
Sure, absolutely. I mean, there's a couple of key factors and differences that I think that are important to understand on that. So on the left-hand side here, you can see that the Flash study. This is the original, again, the positive phase 3 study that we published in JAMA. Here we broke up the treatment cycles into 3 cycles. The main difference that we did there between the cycles was what was treated and with what. The first part of the cycle was a double-blind, placebo-controlled randomized study. Actually, this Flash trial itself is the largest multi-center, double-blind, placebo-controlled randomized study ever conducted in cutaneous T-cell lymphoma. So I'm pretty proud of that to start with. On top of that, it was positive. It was successful. It meets its primary endpoint, so that's always great news for the patients as well, right? In cycle 1, like I said, we selected index lesions. We treated for 6 weeks, and we had treatment breaks. After 6 weeks of treatment, they had a 2-week break, and then we evaluated at week 8. Cycle 2, we crossed over. The difference between cycle 2 and cycle 1 is now it was no longer double-blind, placebo-controlled. It was open label. So everybody's getting drug in cycle 2. But the treatment schedule was exactly the same as cycle 1 and exactly the same as the IIS. It's 2 times a week for 6 weeks. The difference is had the 2-week break after the 6 weeks, and then we evaluated at the end of week 8. And finally, we went into cycle 3, and cycle 3 probably mirrors the IIS the most. And what we did in cycle 3 was it was real-world use. So now we were treating the entire body, not just set index lesions. We were treating any area of disease that the patients and the physicians wanted and were able to treat. But again, 2 times a week for 6 weeks with a 2-week break and evaluation at the end of that 8-week period. So you can see there was a decent amount of starting and stopping. The other thing that's important to understand is that each time we started and stopped in between cycle 1 and cycle 2, we reset back down to the original light dose. Okay, so we went all the way back down. And remember, the light is titrated up during the therapy, just like a lot of other medications. You keep going up and up as you're able to tolerate it. Well, it's kind of similar like that in photodynamic therapy as well. Okay, we were starting and stopping. First off, they were not receiving treatment for those 2 weeks, and we were going back down in dual dose. Now go over to the right side of the slide and see how that compares. If you look at the Flash 2 study, here, again, appropriately so, it is a double-blind, placebo-controlled, randomized clinical trial. But here, we're treating just like we're treating in the IIS. Like I said before, we're treating continuously. We're not having any of those treatment breaks. Okay, there's no start and stop for 2 weeks at a time point. And so the segue of therapy that these patients are getting is basically identical to what Dr. Kim is doing in her investigator-initiated study. Again, that primary endpoint is at week 18. And then we have 3 months follow-up. So because of those factors and the reasons that we know that the FLASH study was already positive, right? We published the results. It met its primary endpoint. And the one thing we learned from that FLASH study was that the response rate continued to increase as we went on, okay? Here now, we're not doing the treatment breaks. We're continuing to treat for 18 weeks. I have every reason to believe that we'll see some positive results out of that. Furthermore, one of the main differences that we have in the confirmatory FLASH-2 study and the original FLASH-1 study goes back to what I was talking about at the leading medical institutions that we interact with over the last decade. These are the primary treatment centers. These are the centers of excellence that are conducting this trial. Every single one of our clinical sites, with the exception of one, was a member of the previous FLASH study. They're known to us. They're familiar with HyBryte. They're excited and engaged to participate in the trial. They were happy with the phase 3 data that we got out of the first study, and they've been supportive. They have buy-in. They helped us with feedback on protocol design. They helped us with interactions with regulatory authorities. They've interacted with us all along the way. And quite frankly, they're somewhat desperate because there's not that much out there for their patient community. So they're very eager for treatment options. And as Dr. Shaber alluded to earlier in the call, one of the things you have to remember about this treatment landscape is that it's a chronic disease. The patients are going to be treated for very long periods of time. So the efficacy, definitely important, and we're certainly showing efficacy. And we're showing efficacy in an 18-week time point, which is very significant. But just as significant is the safety profile, okay? The fact that the patients can tolerate this therapy well, and the fact that there's no DNA damaging side effects from our drug, there's no systemic side effects that we've witnessed from our drug, and the fact that this therapy is, as a whole, extremely well tolerated, it's very important for the patients. And it's something that we're very, very happy to see.
If I can just highlight too, Dr. Poyan, is of something you said earlier, is in the first phase 3 study, the FLASH study, we showed statistical significance in as early as 8 weeks, 6 weeks of treatment. So with the confirmatory study, looking at a longer, which is the FLASH-2, looking at a longer comparative treatment duration out to 18 weeks, I think the IIS, the investigator-initiated study, gives us that peek into what we would expect to see in that confirmatory study that runs very nicely from one to the other. So I think we've really, with the data we've generated to date, not only in the first phase 3, but with the supportive studies like the investigator-initiated study, have all moved in the same direction, the positive direction, to get to the point you mentioned that one thing we know for certain is the longer you treat with this therapy, the better it works. And that's what we've seen in every study we've conducted to date. You apply the HyBryte, the synthetic hypericin, to the lesions topically, you activate it with visible light, and that treatment continues, and you see a shrinkage of those lesions over time.
Absolutely. And finally, one good thing to highlight, I think, in regards to the FLASH-2 study, like I said, that it's ongoing, and I highlighted the safety profile, but also because these centers are all known to us, the enrollment rate. These patients, the physicians, they already had patients in mind when we were getting ready to open their centers up. Okay, so we have every reason to believe that we should be able to enroll in this clinical research study a lot quicker and a lot more expeditiously than we did in the original FLASH study.
That's fantastic. Thank you both for that. Appreciate you reviewing the studies with us, Dr. Pulian, and I think it's very insightful to hear your expert insights on that, as well as hear all the factors of why this FLASH-2 study are, it's coming. It feels very de-risked, right? You've done a previous trial in a similar patient, exact same patient population, but similar study design. You've improved on that study design. You're working with familiar centers of excellence. You have a positive efficacy show, positive safety show. It seems like there's a lot of dominoes that are lining up to make this confirmatory study a great outcome for the Soligenix team. So that's really helpful, and obviously there's a severe unmet need with this disease. So great to get.
I just have to add that there's always risk in drug development, right?
Of course.
I don't want to say there is no risk, right? There is risk, but to your point, the work we've done has really de-risked this program because we understand it better, right? We're building on a lot of data that we've generated over many, many years. And as I noted, that investigator-initiated study that's very similar to the confirmatory FLASH-2 study gives us an added peek into what we would expect to see. So we believe the probability of success is high, but again, I just want to qualify that point.
Yeah, no, appreciate that. And you just did what they do in the best movies, right? You said the title of the movie in your response there, peek into our late stage study, right? So I'm sure that's what everyone was looking for there. But anyways, no, thank you both for those details. I think that was explained very very eloquently and really paints the picture clearly. So as we move ahead here quickly, Dr. Shaber, I know we've talked about the study, and I think people want to understand kind of what's coming up next. So can you provide a little context on what we should expect as far as upcoming corporate milestones for HyBryte and what people should be looking out for?
Sure, I can give you what we've publicly disclosed to date, right? We expect what we said is we expect top line results in the second half of 2026, mid to second half of 2026 next year. Although in 2025, in the second half of this year, so a little bit later, I'll be giving an enrollment update that will give us a sense of where the enrollment is and if that timeline requires adjustment. As Dr. Poulian said, we've enrollment is going better than expected, but I need a couple more months to really see the enrollment trajectory, all the sites up, all the sites active enrolling, to really understand where that enrollment curve is. And then I'm hopeful that I'll be able to provide some further guidance a little bit later this year on that timeline and overall trial.
Fantastic. Yeah, no, that sounds great. So appreciate that that messaging and that highlight of what's to come. So with that, I think we covered most of the content of what we wanted to focus on today. So would love to move into some Q&A. I see a couple questions have come in while we've been on the call here. So I'm going to go ahead and pull those up and ask those to you, both of you, and we can answer those. And we have a little time left, so should be able to get a couple of these done. So let me just pull this up here. Okay, great. So the first question that we received was, how does HyBryte compare to other treatment options that are out there for CTCL?
Dr. Poulian, you want to take that one?
Yeah, sure, I can take that. Okay, so I think, well, actually, the answer to that's probably twofold. First off, and one thing I didn't touch on because I'm trying to be high level with the study design, but in addition to reaching the primary endpoint in the FLASH study, one of the really significant clinical findings that we got out of the original positive phase 3 study was how it worked on different aspects of disease. And what I mean by that is that cutaneous T-cell lymphoma can manifest in a couple of different ways. It can either be patch-like lesions, which are kind of thinner and on the and flat to the skin and diffuse throughout, or it can be plaque-like lesions. Okay, so they're thicker. And again, right, this is a topical treatment. So compared to other topical treatments, a lot of times the available options out there to patients right now are not as successful in treating the tougher, thicker, harder to treat plaque lesions than they are with the patch lesions. We found the exact opposite was true with us in the original FLASH study. We found that our HyBryte worked equally as effective in patch and plaque disease. Okay, now it may be due to the fact that the visible light that we're treating the patients with penetrates deeper into the skin, but regardless of the reason, the data is clear. Those patients have a positive outcome, and these are some of the patients, and that's really significant because to understand, they already have limited options. If the limited topical options that these patients have don't work, then the physicians have no other recourse other than to go to more caustic, systemic therapies. Okay, so think about that. The chemotherapeutic agent that they're giving them topically doesn't work as much. Now we've got to get them something that's a systemic therapy with a higher side effects profile. And that's certainly not advantageous for the patient. So the potential for HyBryte to be equally effective in all manners of that disease is definitely a differentiator in that regard. I think the other thing I would highlight again is the safety profile. I mean, I mentioned that a lot of what's available out there that's approved anyway is really a topical chemotherapeutic agent. We're a topical therapy as well, but we're not DNA damaging. Okay, we don't have any immunogenic effects. We are not very well systemically absorbed at all. I mean, we see extremely minimal concentration in the bloodstream, and we have to go down to extremely small detection limits. So it's practically not systemically absorbed at all, which means that you don't see any of those like normal side effects that you would see with other systemic therapies. If you think about it from a patient point of view, like Dr. Shaber said before, they're going to be on therapy for quite a long time. If the agent that you're taking, like for example, like meclorethamine gel, which is one of the options that works for some patients out there, is so caustic sometimes that 20% of their patients can't continue on to the therapy, or if they are doing well with the therapy, they can't expose it to any visible skin, any other non-disease skin or partner skin. I mean, that means these patients are taking something to treat their cancer that means that they can't, share a bed with their spouse or give a hug to their kid. That's significant, right? We have an option here in HyBryte that is topically applied. It does not have any side effects profile that's appreciable anywhere near the magnitude of what we're seeing that's out there right now in the market. So, I mean, you ask me why, how is it different than what's out there? I would tell you that, in my opinion, I think it is a safer, more realistic option for a patient to be on for their disease.
That's fantastic. Thank you. Dr. Shaver, anything you want to add to that, or did Dr. Pullian cover most of it?
No, Dr. Pullian covered most. I would just say that, again, our focus is early stage disease and a frontline therapy. And that's why it's so important. As you would imagine, when you're embarking on this lifelong disease, you want the safest option first that's the most effective, right? You don't want to go into the DNA damaging types of therapies. And we believe that HyBryte provides that first option that I think many of the clinicians, the key opinion leaders we work with, really are embracing here. That's why they're working with us like they are in completing this confirmatory study.
Fantastic. Yeah, no, that's like you said, extremely important. So love to hear that. We probably have time for one more question. So I got one here that I think is that's important to address given the rare disease status. But the question is, what is the revenue potential of for HyBryte?
So in our corporate presentation, we've disclosed that we said worldwide, it's in excess of $250 million in the U.S. At peak sales. We're probably looking approaching $100 million. So while it's an orphan, a rare disease that we're treating, it does have a significant market potential, especially when you think about that frontline or early stage treatment of those mycosis fungoides patients, which make up the largest percentage of cutaneous T-cell lymphoma.
Gotcha. Yeah, no, that's super helpful, and I'm glad you clarified that point, because I think a lot of folks associate kind of the rare disease or their orphan drug nation clinical development as like not as large of markets, but there actually are quite substantial revenue potential for these diseases because there is such an unmet need. So it's very important, and there's definitely a commercial opportunity there. So we've got a couple more questions, but unfortunately we're up against the time. So I think what we'll do is for those who submit these questions, we have your contact information. So we'll follow up directly with answers to those questions to make sure that those get addressed. And there's some good questions in here about kind of other aspects to the pipeline and what's coming up. We'll be doing more topics, like more events like this and content to push that out. So would invite you to follow along with the story to see those. But in the meantime, I want to thank everyone again for their time. And Dr. Shaver, Dr. Pullian, I'll turn it over to you guys for any kind of closing remarks you have before wrapping up here.
Very, very briefly, again, thank you for having us. For those that are interested and want to learn more, we have a lot of good information on our corporate website, including some publications of our previous studies that are always helpful. And so you can find that information there and quite a bit of additional information about not only HyBryte, but other programs that we have ongoing as well.
Fantastic. Well, thank you again, both. Appreciate the time, and look forward to touch base again soon.
Thank you.